Hello everybody, and a warm welcome to this first appointment of the medical breaking news. The aim of this program is to offer an updated follow-up to the most prominent international congress about the main issues of medicine our age. I am a journalist, Antonio Caprarica, and as you may know, in our line of business, the old saying goes, good news, no news. Well, we are going to prove the contrary because we have got a lot of good news today, and we are going to discuss about that with our two guests here in the studio, a lot of others out of this studio, but here we have Luca Revelli, surgeon at the Università Cattolica in Rome, and Professor Mario Airoldi, who is the head of Oncologia Medica 2 Ospedale Molinette here in Turin. Now, let's go back to the first Congress we are going to talk about. It was held in Washington, devoted to resolving cancer heterogeneity. What was that about? Well, the intratumoral heterogeneity is a key perspective from many point of view, mostly for therapy. Why? For instance, two patients, same type of tumor, the same level of staging. Why? One reacts very well to the therapy and the other, unfortunately, does not. This is one of the issues we are going to face today. Also, the Congress in Washington analyzed the relation between the intratumoral heterogeneity and the growth of metastases. It paid a specific attention to the so-called liquid biopsy with the cellular plasticity, highlighting the complexity of a diagnostic and therapeutic approach. Also, in relation to the liquid biopsy, a large relevance has been given to the circulating tumor cells, CTCs. They are considered a very useful tool for the evaluation of the tumor, its likely developments, and obviously the therapeutic implication. Finally, the last session, a whole session has been devoted to the therapy in relation with the tumoral heterogeneity. Now, we are, before starting the discussion here in the studio, we are going straight to Washington, where there was for us our colleague, Dan Keller. Hi, Dan, how are you doing there?
Thanks, Antonio. I'm here in Washington, D.C. at the International Symposium on Resolving Cancer Heterogeneity, where world-renowned experts came together to present their data. One person I spoke with was the president of the symposium, Dr. Ignacio Wastuba. And I also spoke with Dr. Andrew Futrall from MD Anderson Cancer Center in Houston, Texas. Have you seen updates coming out compared to last year's meeting or different topics?
Role of immune response in cancer development, progression, and the role on also resistant to therapy. And most importantly, because we are now understanding the biology of the immune response in cancer, there are new ways to modulate that, and there is immunotherapy now is available for our patients that show success in certain tumor types, but also that has an intrinsic intratumoral heterogeneity characteristic that actually ties to the intratumoral heterogeneity genomic of the tumor too. So that is the new area.
This gives you a lot of data. You get essentially a constellation here. How do you make sense of it, especially if you have conflicting kinds of things within a tumor?
I mean, I think you hit the crux of the matter. I mean, I think we are working in baby steps at the moment in terms of really thinking about how to get to grips with these integrated type analyses. We tend to take each data type, burn it to the ground, and then come up with some truths and then try to sit back and weave the truths together. And that works to a certain extent, and it's worked pretty well for molecularly targeted therapy, things like kinase inhibitors where we have very specific activity. When you move to something like immunotherapy where you're hitting a complete complex ecosystem, I think it's falling short.
Mario Airoldi, I'd like to have your comment, your opinion on the interviews we have just listened to.
I think that immunotherapy and target therapy are the future of oncology, but we need to know the responders and the resistant patients. Resistance is due to cancer cell mutation, and many mutations are now druggable, and we can observe clinical response with this new drug according to the mutation of the cancer cell.
Luca Revelli, whom I already introduced to you, was there in Washington, and obviously we take advantage of what you have brought back here in your bucket. Could you show us something interesting?
Yeah. You see, Antonio, I found something particularly interesting about brain tumors, about angiogenesis on liver cancer, and about genetic changes of T cells in the blood.
So it's becoming more and more clear and obvious that the tumors, even if they look the same, and even if they're driven by the same genetic changes, they're actually not created equal. And tumors carrying exactly the same genetic mutation, such as BRV600E, will behave very differently, whether they're in a brain tumor or they're in a skin tumor. And this is sort of a different type of a spectrum, different type of heterogeneity. So while the original idea was that we will just sequence the tumors and based on mutation, we will know how to treat them, we will know how they behave. Now we know that we actually really need to have a context of a tissue and age.
So since the early 1970s, when another surgeon, Judith Folkman, published a pivotal paper where he showed that as tumors grew, they needed to generate new blood vessels, and that's called angiogenesis. And since that time, it's been like the holy grail of vasculature for tumors. And all the drugs that we have developed have been targeting that pathway, the angiogenesis pathway. Unfortunately, for the last 30, 40 years, it has not panned out. We have seen a modest effect of these drugs, despite developing many of them. And it was a little bit puzzling. And what we've discovered in our tumor group, which is colon cancer liver metastasis, I use it as a model because we actually operate on many of these patients, so we have access to the tissues, is that yes, a group of them do have angiogenesis and develop new blood vessels in order to feed themselves. And those, if you give them anti-angiogenic drugs, actually is very effective for them.
This means that the eukaryotic cell, which is different from the prokaryotic cell, can do many different variants with functional differences from the same gene. This is an advantage in terms of evolution. And for normal cells, in a physiological terms, it enables, for example, adaptation to different media. It enables development. It enables economy and versatility. However, things can go wrong. So splicing could be a mechanism adding to the already complex field of cancer, because if something goes wrong with splicing, you can end up in a cell with a variant that is doing what it's not supposed to do.
In Washington, there was also Sergio Abrignani, a very famous Italian scientist. He is the director of the National Institute for Molecular Genetics. I think it's very interesting to listen to his interview, which has been given to our friend Dan Keller.
You were focusing somewhat on regulatory T cells, Tregs. What's your strategy there?
Our strategy has been to identify some cell surface markers that are specifically expressed on Tregs in the tumor and not in Tregs all over the body. Whereas so far, all the immunotherapeutic approaches have focused on monoclonal antibodies specific for molecules that are present on all Tregs. So we have identified the signature, which means molecules selectively present on Tregs in the tumor, toward which we are making monoclonal antibodies with the idea of developing new therapeutic monoclonal antibodies that at that point should eliminate only Tregs in the tumor and not Tregs in other part of the body, therefore avoiding autoimmunity, which is the complication when you avoid, if you eliminate T cells all over the body.
Do you have monoclonals or any other agents that you are?
We're trying to develop monoclonal together with the pharma company, monoclonal antibodies capable of recognizing a series of targets we have identified, and we hope in 2.5 years to go into clinical trials. This is the key message. The other key message is that we should look at intratumoral T cells and tumoral T cells together. I mean, it is the notorious cancer immunoediting, that is understanding how the cancer cells are influencing the immune cells and vice versa, immune cells are influencing the cancer cells. If we can understand this, we may come up with new therapeutic targets.
Mario Eroldi, which kind of drugs we can rely on up to now for the immunotherapy, and which kind of expectation would we have for tomorrow?
Now we use the checkpoint inhibitors, but we know we have some cells like macrophage, neutrophils, Tregs, that can induce resistance to immunotherapy. The future is the combination of immunomodulating drugs, the combination with chemotherapy, radiotherapy, and target therapy. And the next future is the use of immune cell as a drug.
Well, I'd like now to show you the interview which we have got with another fellow countryman who works in Australia. Is Dr. Riccardo Dolcetti with the University of Queensland. Please, let's see it.
What are you doing that is going to give you widespread applicability here and more efficacy?
Yeah, we know now why in the previous clinical experiences vaccines failed, because as a single therapy they are very weak in terms of clinical benefit. Now we know how to combine the vaccines with the new drugs which are able to unleash the T cells which are present in the microenvironment. So if we combine a strategy as ours, able to induce new forms of T cells able to recognize antigen with other strategies which are able to relieve the immune suppression of the tumor, we hope to be able to overcome the current limitation and improve the response rate of the patients. Our platform is able to avoid all these complex and expensive steps, as it is able to directly target the tumor antigens in vivo, avoiding any ex vivo step.
Mario, the point is that vaccines in the past failed. So what could be now the way forward?
Now the future is the use of vaccine in combination with immune drugs or immune cell to induce and give an enhanced of the immune response.
Luca, what else have you got in your baggage? Show us something else very interesting.
Yes, Antonio, I'll find something really fascinating. For example, is it difficult to find cancer cells in the blood, the so-called CTC cells?
There are. For example, we were speaking today about a lot of new things like so-called liquid biopsies. So you can have to study tumor DNA in the blood of the patient. And we still use that just as a research. I'm pretty convinced that we have to take it directly to clinics and to have a direct experience in the patients.
It's very difficult to find these EMT cells, EMT cancer cells in the blood, because they have then, in theory, a lot of mesenchymal features. Problem is that the way how we right now try to find these rare CTCs is actually by their epithelial phenotype. So if a cell is in a fully EMT state, that would mean that we couldn't actually find it by the technologies that we use.
Cancer in a patient, but then this cancer cell and the population that comes out from this cancer cell has to crosstalk with the environment. And so this has to do with the response of the patient to the cancer. So understanding the cell composition, which kind of cells, which kind of molecules are interacting with this cancer cell in each single patient is the key, actually, to understand and to hit the real stem of the cancer for each patient.
Well, these are the issues Professor Alix Panabier in France and Professor Klaus Pantel in Germany mostly work on. I think it's extremely interesting to listen to their interviews now.
What can circulating tumor cells tell us after initiation of therapy?
It's very important to study the kinetics of CTCs, mostly after the initiation of therapy, because you have quick information on responders to these specific therapies, whether you have a stable or increasing or decreasing number of CTCs very quickly, and maybe we are able to change the therapy.
Besides looking at numbers, what other things can you detect by looking at single CTCs?
So we can enumerate them, but it's very important to characterize them as well at the genomic level, transcriptomic level, proteomic, secretomic levels. It is really the plus of the circulating tumor cells.
Can you also detect whether they have metastatic potential and prognosticate based on that?
Yes, we work now for many years on metastasis competent CTCs. They are really a very small subpopulation of CTCs, and they are the ones that are able to give rise to metastases at distant sites. So it's very important to understand them, to be able to eradicate them and to...
Can you tell us about what you have planned for this conference next year?
Well, I think next year we will probably have the conference in Berlin, and we will focus a bit more on the different possibilities to do liquid biopsy. Besides CTCs, there are also DNA and exosomes that could contribute and complementary information to individual patients' treatment. And it might be nice to see how these really different ways to do liquid biopsies interact, complement each other, or are redundant.
Mario, the CTCs are not actually now the only biomarker we can rely upon. What else we can expect for the next future?
In the next future, I think we can use also DNA or RNA circulating in the blood, or the use of exosomes.
Well, thank you very much to Luca Ravelli and Mario Airoldi. I really look forward to seeing you again very soon. And I wanted to thank mostly our friend Dan in Washington and to tell him also goodbye very soon.
Thanks, Antonio. We're going to be leaving this conference and bringing you more conferences from around the world with important scientific information in the coming weeks and months. From Washington, D.C., I'm Dan Keller.
Well, there is only one thing I still can do, and just thank you for your attention and tell you also goodbye. Where and what about, I don't know yet, but you can start guess. Bye-bye.